Hello, and thank you for joining us at Track 4, Science and Technology Innovation, sponsored by PRA Health Sciences. I am Jana Drummond. I will be your host for today's session on rare research resources, biomarkers. As a reminder, this track is being live streamed, and I would like to welcome our remote attendees and remind everyone to download our mobile app to use some of our highlighted features like the live Q&A. Understanding how biomarkers can be used in research and development can be key to diagnosing or assessing the progression of rare disease. Because of this, it is important to learn how biomarkers are identified, validated, and the critical role it can play. To discuss the topic further, we have Stephen Maricich, Medical Director at BioMarin Pharmaceuticals, and Holly Kordasiewicz, Executive Director of the Neuroscience Drug Discovery and Biomarker Developments at Ionis Pharmaceuticals. Let's welcome them up.
All right, well, thank you for that introduction. I want to thank the organizers for inviting me here today. You know, as being in drug development or therapeutic development now, we don't get that many opportunities to actually talk to patients and their families and advocates, and so it's a really special thing, and I'm really pleased to be here and be part of this meeting. So I've been tasked today with introducing the concept of biomarkers. Before starting that, though, I want to take a step back and talk about the thing that we're really focused on in clinical development, which are endpoints. So an endpoint is anything that can be measured and analyzed in a clinical trial. And endpoints come in a couple different flavors. The ones that we really care about or that we're trying to move with the different therapeutic interventions that we develop are clinically meaningful endpoints. And so what does that mean? A clinically meaningful endpoint is something that directly measures some aspect of the pathophysiological process that is involved in a disease. Now, one very big piece of this is that these endpoints have to be important to patients. And so if you think about clinically meaningful endpoints as somehow measuring what a patient feels or functions or survives, that'll give you the idea of the group of things that we're talking about. So examples might be survival in cancer, stroke and heart disease, or pain and arthritis. Now, there are certain times when it's very difficult, and we'll talk about this a little bit in more detail in a little while, where it's very difficult to either identify or to measure these clinically meaningful endpoints. And in those situations, you might want to identify a surrogate endpoint which can serve as a substitute for that clinically meaningful endpoint. And in general, if you've got a good surrogate, it is going to reflect changes in the clinically meaningful endpoint itself. So how do you know that you've got a good surrogate, or what are the key features of a good surrogate? Well, first, it's got to correlate in a reliable and a reproducible way with the clinical endpoint that you care about. And examples of this might be systolic blood pressure as a surrogate endpoint, and its correlation with stroke occurrence, which is the clinically meaningful endpoint that has been the endpoint of a number of trials. But equally, or if not more important, to decide whether or not you have a valid surrogate is it has to fully capture the net effect of the intervention on that clinically meaningful endpoint that you care about. And in general, if you can develop something that's going to track both beneficial and negative effects of your treatment, that's going to be a much more powerful predictor than if you can't do that. Now, the way that you do that in practice takes a lot of patience. Ideally, you do that over a lot of different trials so that you can see how reproducible this data is that suggests that what you're looking at in the surrogate is predictive in some way. And when you've been able to show that in a convincing manner, then you can say that that surrogate endpoint is in fact validated. It's a valid predictor of your clinical endpoint of interest. And this is easier to do if you understand in some detail the pathophysiology of the disease process, and equally importantly, if you understand the mechanism of action of your therapeutic intervention. So what does a valid surrogate endpoint look like? This is an example that's taken from the literature. The disease here is stage 3 colon cancer. The intervention is the use of 5-fluorouracil, which is an adjuvant chemotherapy that's used for treatment. The clinical endpoint we care about is overall survival in this disease, and the surrogate is going to be 3-year disease-free survival. And what's graphed on the right is data from 18 different studies where on the x-axis you're looking at hazard ratios from disease-free survival, surrogate endpoint, and on the y-axis you're looking at the overall survival as the clinical endpoint. And what you can see is that when you plot out what the disease-free survival was predicting would be the overall survival, across all these 18 studies you can basically draw a straight line. So this surrogate has very strong predictive power that leads you into some insight on the clinically meaningful endpoint. Now, again, we're going to take another step back here and talk about biomarkers, because surrogate endpoints are in fact a subset of the broader group of biomarkers. So what is a biomarker, a biological marker? It's any measurement of a biological process. So these can be physiological, they can be things like blood pressure, heart rate. They can be biochemical, cholesterol, blood sugars, genetic or metabolic if you're looking at gene sequences, enzyme levels, or any measurements that you can get from imaging, x-rays, MRIs, CT scans. Now, biomarkers can be useful as surrogate endpoints if they satisfy the following criteria. First, the test that measures a biomarker has to be reliable and reproducible, and you also want it to be sensitive and specific so that you have a sense that the data that you're getting from that is going to be good and valid data. Secondly, again, you want it to be correlated with the clinically meaningful endpoint or outcome of interest. And then it has to be appropriate for the proposed use. So an example of this would be the use of hemoglobin A1C as a biochemical surrogate, which has been shown now to be predictive of the risk of microvascular complications in diabetes, which is your clinically meaningful endpoint. Okay. So if the gold standard is to identify and measure clinically meaningful endpoints, why do we even care about biomarkers or surrogates? When are they useful? Well, the main reason that they're in use today really comes from this overall goal, which started in the 90s, of being able to provide patients earlier access to promising treatments. Because as I'm sure everyone in this room is aware, the drug development process or the therapy development process is a very long and drawn out process. It starts with a scientific idea, moves through preclinical testing, finally gets into the patient arena, and then has to undergo, under normal drug development, multiple studies to be able to show both therapeutic effect and safety. And this can often take 10, 15, or even more years to go from point A to point Z. So the benefit of then using biomarkers or surrogate endpoints is that they can shorten study time. These clinically meaningful endpoints, if you're looking at things like that take a long time to occur, like death from a slowly progressive disease, or things that happen at unpredictable intervals, like heart attacks, can be very difficult to enroll and then follow a clinical cohort for long enough to be able to see those clinically meaningful endpoints. If you've got a predictive biomarker or surrogate, you can do it on a much faster time scale. And not only does that shorten the time, but it ends up costing less in terms of trial development. And then secondly, you can include fewer people in your studies. This is really important for rare disease. We'll come back to that right at the end of the talk. And again, this can reduce cost. And then finally, another important aspect that's often overlooked is that you can sometimes get early reads on the safety of a therapy if you can somehow correlate that with your biomarker of interest. So there are a lot of advantages then to using surrogates and biomarkers when you're able to do so. There are also now dedicated regulatory pathways that take this into account when one is evaluating and trying to get new therapies approved. So both the FDA in the form of accelerated approval and the EMA, the European equivalent, in the form of conditional approval, have these pathways. And these really allow marketing of therapies that have proven biological effects on the basis of biomarkers or surrogates, but may not at the time of approval have proven effects on the clinically meaningful endpoints. The FDA has a list of 4 things that they require for you to be able to satisfy this criteria and be considered for accelerated approval. The first is that the disease has to be either life-threatening or induce irreversible morbidity. And the reason for that is a benefit-risk assessment. So one can imagine that if you're shortening the drug development time or the therapeutic development time, you're not going to have the same safety readout as one might have from a much longer study. So here, if you start with diseases where not only are they horrible diseases, but then there's also the second point, which is a substantially unmet medical need. There's no effective therapies that are available. You can skew that risk-benefit axis more toward benefit. Another important piece of this is that the surrogate endpoint should reasonably predict the clinically meaningful response. And that language is actually very important because we talked about how you validate a surrogate endpoint and that you have to have multiple studies, lots of people, multiple demonstrations. What the FDA is saying here is reasonably likely to predict. That bar is a little bit lower. And again, this is good for rare diseases because it's very difficult to have multiple studies with lots of patients if you only have 100 people in the world that have a disease. And then finally, there's a requirement for a confirmatory study to demonstrate that you actually have some clinically meaningful benefit that is associated with your surrogate endpoints. Now, if you have a validated endpoint, that endpoint can actually serve as the full basis for full approval without the need to assess clinically meaningful endpoints. However, when you think about it, that relationship between the validated surrogate and those clinically meaningful endpoints would have already been established in multiple other studies. So in aggregate, to date, about 92 adult and 56 pediatric indications have used or have been based solely on the use of surrogate endpoints for FDA approval. And in fact, interestingly, between 2010 and 2012, about 45% of all approvals either cited the use of or were based solely on the use of surrogates. And if you look also at the population, about 20% of them actually took advantage of this accelerated approval pathway. So there's a lot of talk now about biomarkers and surrogate endpoint use in therapeutic development. Those are all the good things about biomarkers and surrogates. What are some of the challenges that we have to think about when we're trying to use those as part of our development plans? Well, one of them comes down to this issue of, is your endpoint, your surrogate, truly predictive, or is it merely correlative with the clinical endpoints that you care about? Ideally, what you want is your surrogate to be directly in that pathway, the disease pathophysiological pathway that lies between the disease itself and the clinically meaningful endpoint of interest. Often what you end up with is this second situation where the disease may act both on the pathway that includes your biomarker and on the clinically meaningful endpoint, but those things are parallel to one another. And you're left with a question of whether or not that biomarker is really truly representing every single piece of the clinical puzzle. The bright side is that in these situations, although you might not be able to use that particular surrogate for a drug approval, those surrogates may still be important for either diagnosis or prognosis. So for example, prostate-specific antigen is a great example of this, where the predictive power of you developing prostate cancer by looking at PSA levels is very low, but as a monitor for the reoccurrence of cancer in individuals who's already had it is actually much better. Secondly, you want to know how your surrogate tracks with safety. And so as we compress the clinical development timeline, because we're using a surrogate rather than that clinically meaningful endpoint that we want to look at, what one hopes is that any adverse events that you see in that shortened timeline are going to be representative of events that are going to happen later. What you want to avoid is a situation where a whole bunch of new adverse events come up at that later stage after patients had been on a therapy for longer. And this is actually a major reason why both FDA and EMA require confirmatory studies. It gives us more of a sense of the overall safety paradigm for any new treatment that you're thinking about. And then finally, there's this question of universality. So it may be that your surrogate or your biomarker is valid in a single disease state or with a class of therapeutic compounds that work by a certain mechanism. It may not be true that you can universalize that to other disease states or other classes of medications. And therefore, it would require more work to convince yourselves and others that you're onto something in terms of predictive power. Now, I want to leave you with some thoughts about how biomarkers and surrogates can be used and are important for rare disease. There are many factors that actually support the use of biomarkers and surrogates in rare disease research. First of all, most rare diseases have a very clear single genetic or metabolic cause. And therefore, that suggests plausible biomarkers that one should be able to substantiate through preclinical testing and through biochemical testing in patients. Typically, there are small affected populations, and so these surrogate endpoints can drastically reduce the trial size that is necessary to be able to show some sort of beneficial effect. The lack of prior natural history studies in rare disease populations often leads to an incomplete understanding of the clinical manifestations of the disease, and therefore it can be difficult to identify clinically meaningful endpoints. And so if you have a good biomarker, you can use that as a proxy as you're gathering more information about the disease to see whether or not your therapy is making a difference. Most of these diseases, or at least many of them, are slowly progressive and they have low event rates. So again, in a short period of time that is conducive to clinical development, it's difficult to show clinical responses. And then finally, and perhaps most importantly, there are often delays between when irreversible damage starts to take place in these diseases and when patients are ultimately diagnosed. I can tell you, I'm the clinical head for the Sanfilippo syndrome type B program at BioMarin. It's very true in this disease. Parents will tell you that their children become symptomatic between the ages of 1 and 2 years of age. The average age of diagnosis is closer to 6. So we've lost a lot of time when we could have some sort of therapeutic intervention and actually make a bigger difference maybe in these kids' lives. If you've got a good biomarker that tracks with the damage as it's occurring, you may actually be able to see that damage taking place earlier than if you wait for the clinical endpoints. Now, the challenge with rare disease research is that it's really tough to validate these biomarkers. And the FDA recognized this, and in 2012, in the FDA Safety and Innovation Act, they really took this into account, and they gave more broad latitude to the directors at FDA, and they loosened some of the criteria that had been used historically for accelerated approval as it relates to biomarkers and surrogates. And so the development path is still open, and now it's a more holistic approach that looks at diseases on a case-by-case basis, therapies that's being proposed, the biomarkers themselves, and takes into account both preclinical and clinical data to be able to accept these biomarkers. So just in summary, surrogate endpoints are substitutes for clinically meaningful endpoints. These biomarkers, many of which can be used as surrogates, measure physiological processes and can be used, hopefully, as surrogates. The challenge, though, is to make sure that your biomarker or your surrogate actually predicts what it is that you are hoping that it's going to predict. There are regulatory pathways that are in place that can take advantage of these mechanisms for development. And overall, when used appropriately, both biomarkers and surrogates can really shorten therapeutic development time, and they stand a chance to bring effective therapies to patients sooner, which I know everybody in this room, me included, would really like to see. So if you guys will hold your questions, I'm going to introduce Holly, and she is going to apply some of these principles to her own work at Ionis and tell you how they can be used.
Thank you. Perfect. Thank you so much for giving me the chance to speak to you today. I'm really excited to be here. It's been a wonderful meeting so far. And so we've heard a lovely summary of biomarkers, what they are, and the value of them and how they can be used. And so what I'd like to do now is walk you through a real-life example of a drug development process from start to finish. Well, we're not quite to finish yet, but from start to a successful phase 1 trial. And the example I'm going to use is our Huntington's Disease program. And here I'll highlight the different steps where the biomarkers fit into that process and what that ultimately means for how we can accelerate this program and how we are accelerating this program. Okay, so our Huntington's story is a story that I particularly love. It's a story that it's not finished being written yet, so it's still an ongoing program, but it's a program where the Huntington's disease community has really gotten together to do the foundational science. So understand biomarkers, understand endpoints, understand how to quantify the disease, so that to really enable clinical trials, so that as companies are ready with drugs to take into clinical trials, that information is there. And because of this, we've been able to accelerate our Huntington program. And I also think it's a lovely example of some of the things that we've heard about earlier today, how different groups working in parallel with different expertise and different topics can really all come together to result in a successful trial and hopefully result in a drug that's ultimately going to make a real meaningful difference for these patients. So real quick, just about myself and Ionis. So we are a company that focuses on RNA therapeutics and specifically antisense oligonucleotides, or ASOs. We have a large focus in neurodegenerative diseases, including neurodegenerative diseases in the rare space. And so Huntington's is the program that I'll mention today. And Huntington's disease, when we started this program, we didn't know much about it. So we knew it was caused by a CAG expansion in the Huntington gene. We thought that this was largely led to a dominant gain of function, so a toxic protein, and our approach was really simple. You get rid of that toxic protein, you'll get rid of the disease. We also knew at the time that oligos could work in the brain, but that's about it. We knew it was a horrible disease, we knew it needed help, but there was a lot of work to do. Okay, so there was a number of key questions that needed to be answered when the program started. And a lot of these centered around real basic drug discovery things. So can we get to oligos to where they need to go? Can we really engage our target? Can we have a benefit in a disease model that already has established disease? How do we do that? And so these technology-based questions we were focusing on to try to find the very best drug to take into the clinic. And now while we were doing this, the Huntington's community was also focusing on the foundational science. And this is just as important and critically important to finding that particular molecule to take in to help patients, but also once you have that molecule, how do you know that it's doing what it's supposed to be doing and that it's really working? And so this gets into clinical trial design and strategy and where biomarkers can have a real dramatic impact. And so first, the question is there a biomarker that we can use to make sure our drug, so our oligonucleotide, is really lowering Huntington's, engaging the target in the brain and doing what we think it should be doing. The second question is what endpoints can we look at that we can look at are gonna change the fastest so that we can do the shortest trials possible to get this drugs to patients as quickly as possible. Okay, so a real quick reminder about clinical trial design here. So this is the typical path that drugs take through the clinic. So we have our phase 1, which is really safety, our phase 2, which is the drug safe again still, and then also trying to select, do we have the right dose, and then starting to understand about what endpoints we can measure. And then your final pivotal trial is really, is the drug really safe, and does it have a difference in the clinic? Is it really altering the disease course? And so the idea and the whole concept of biomarkers, which we just heard about, was really to try to make this process shorter, to try to accelerate that as much as we can. And so to do that, we try to answer these questions preclinically and get more information from each phase of these trials to accelerate this process. And so that's what I'll talk to you today about, is exactly how that fit into our Huntington program. Okay, so go way back to the beginning. So when we first decided we were going to work on Huntington's disease, this is work that started in collaboration with Down Cleveland, actually down the street at UCSD. First thing was to make sure Huntington oligos are active in cells. Once we know they're active in cells, we then want to look and see if they're active in models. So just real quick, how oligos work. You have your gene. In Huntington's disease, that makes an RNA that contains the mutation. That RNA is then translated into a protein. That protein is toxic, and that leads to disease. The oligos bind to that RNA. If you get rid of the RNA, you get rid of the protein, and hopefully the idea is that then you would get rid of the disease. And so this is just to show in animal models, we can get rid of that RNA, and then we can get rid of that protein. Okay. The next step was, now that we can do that in an animal model, can we have a benefit on disease? So we went into a number of different animal models, and these are doing different tasks, some of the ones that you've heard about earlier. And looking in those different tasks, when we treat them with our drug, do they have a benefit? And they do. And so we now have a drug that we think is working, we think we're having a benefit, but then there's quite a bit more work that went into really understanding when in disease we should intervene, how much we need to lower Huntington, and then also how to deliver our drugs to the larger brain. And so once we optimized ASO delivery to the larger brain, the next question was, is that RNA and protein really down in the larger brain? And so that's what I'm showing you here is, again, you're looking at the RNA and protein levels. So they're normally at 100%. After they get to therapy, they're reduced. So because these protein levels are reduced, hopefully this is the toxic protein, and then this will lead to a benefit in disease. So this is great. We can do all this in animal models, but the question is, we now have this data in an animal model, but how are we ever going to know in a human patient that Huntington is really down in the brain? So here, we're able to measure it directly. In a patient, we're not going to be able to do that. And so that's where biomarkers become critically important. So one of our first biomarkers we need to look at is looking at that mutant toxic Huntington protein in a source that we can actually get safely from human patients. And clearly, that isn't taking pieces of people's brains. Okay, so this isn't where the Huntington's community, and everyone has been involved through a lot of this process, but this is where some really amazing things happened. And that question of, is mutant Huntington detectable in a fluid that we can look at, was one that was really taken on by the researchers and clinicians, and also particularly CHDI and specifically Doug McDonald, which is a nonprofit organization that's working for a cure for Huntington's disease. And here they went on a campaign to really try to find, can they measure Huntington in fluids that we can assess in patients? Now, one thing I haven't told you about antisense oligonucleotides is you can't take them as a pill. They don't get into the brain, they don't directly cross the blood-brain barrier. So to get into the brain, we just deliver them to the cerebral spinal fluid by a lumbar puncture injection. They go into the fluid that bathes the brain and spinal cord, and then they're taken up into the tissues. So this is great for delivering the drug. We can get good delivery. We can do this at infrequent intervals. In the Huntington program, we did this once a month. But as you can imagine, for a biomarker, you're limited because Huntington is expressed through the whole body. So if we take blood, we're not going to see just what's happening in the brain. So what we do then is we take that cerebral spinal fluid, so we inject a little drug, but then we take a little of that cerebral spinal fluid and measure our Huntington protein. Now, when we started this program, we had no idea if Huntington would be detectable in that fluid. Nobody knew. The whole community didn't know. And so that's the work that was really one of the next game changers in the program. And the game changer was because the scientists from CHDI and the clinicians who were working on this found mutant Huntington in the CSF. They found mutant Huntington in the CSF, and if you look in the control, pre-manifest, or manifest, as the disease got worse in patients, the Huntington levels went up. And then they reproduced this across multiple cohorts from multiple doctors, and they really see that this Huntington is going up with disease. So now Huntington is measurable in patients in a fluid that we think we're going to have an effect in. So now the next question from your biomarker, and again, this was discussed earlier, is really, is it reliably measurable? Is this assay valid? And so this is work now where our colleagues from Roche, so our Huntington's program is partnered with Roche Pharmaceuticals, and they went through and did a bunch of really lovely work making sure that the assay was measuring exactly what we thought it was measuring and that it was very precise and very quantifiable. And this sounds like some of the sort of boring work that goes into some of the science, but it's really important because you really need to know that what you're looking at is really what you're looking at. So now we have an assay. We can measure it in patient CSF. We think we can measure it very reliably based on all the work that was done to validate the assay. But what does that mean? We're going to measure this in the trial. How should this move? Is this really going to move with our drug that lowers the protein in the brain? So we go back to the preclinical species. And so here is again where the monkey work comes in handy. And the reason we need to do this monkey work is we need to build these preclinical models. And the reason we want to do this preclinically is because we want to take a system where we can measure the tissue levels, we can measure the brain levels of Huntington, we can measure the cerebral spinal fluid levels of our biomarker, and we can see how those correlate. And then we can build a model to say, since in patients we only get that CSF level, this is what it means for the brain. So we use this to both set dose and to be able to understand our data that we get from clinical trials and make assumptions about what that means for what's really happening in the brain, which is what we care about. We ultimately don't care about the CSF, we care about lowering Huntington in the brain because that's where we're gonna help patients and we're gonna help this disease by getting rid of that bad protein. Okay, so we do a series of studies in animals, and this is again in non-human primates who are receiving a drug that lowers their Huntington. And the graph that we're looking at is as you increase the dose levels, the Huntington goes down in the tissue. This is the RNA, and it stays down over time. So we're also looking at the kinetics of how this happens. So then we also look at the protein levels in the tissue and see that those are down as well. And then we look at the protein levels in the CSF and see how those are down. And so this is, again, one of those next sort of aha moments in the program where we have a marker and it's actually moving like we think it should be moving. So this is the next exciting point for our biomarker work. So then we went through and took all this data and built a model. And so I can't explain all the math that went into it, but modelers built this and said, this is the dose that you need to give your Huntington's patients, and this is what their CSF Huntington is going to look like, and this is what it means for those levels in the tissues. All right, so it is now 2014, getting ready for 2015, and we're getting ready for our first clinical trial. So we now have a drug. We have a drug that is safe. We have a drug that's effective in animal models. We have a biomarker that lets us know we think we know what to do, and we've picked a dose that we think is going to be effective in lowering this protein. So now we go into the clinic, and this gets us into our phase 1 trial. So phase 1 trials are really safety trials. You're looking at safety of the drug. But here we also wanted to look at, of course, first and foremost, safety, but also what is that biomarker doing? Is our drug really lowering huntingtin in the brain like we think it is based on that mutant huntingtin in the CSF? And so this is a summary of the trial design. It was a placebo-controlled, so patients either 1 in 4 patients received placebo, the other 3 received drug. They received injections once a month for 4 months. And at each one of those times when we injected the drug, we drew a little bit of CSF that we kept. And so the CSF that we kept, we have this baseline sample, and then we have the other samples 1 month later after the dose. And that's where we can look at the huntingtin and see what has happened to that huntingtin protein over time. This is exactly what we had done in the non-human primates, so we have a model for what we think this should look like. Okay. We also have a time point that is a little bit later to see, is this really staying down for a longer period of time? Okay. So now we take these samples, and we will look at them for our mutant huntingtin, and this is just one more summary just to make sure that everybody understands the trial design, is we go through and we have different cohorts, so a different group of patients either got dose A, B, C, or D, and we increase the doses over time, and as we increase the dose for each one of the patients, we end up with groups of patients that received either a low dose or a high dose. And then we look at the huntingtin protein, and this is one of the most exciting graphs. I can't even explain how it was when everybody finally saw the data. And here is what you're looking at is huntingtin protein in the CSF from the patients who received the drug. So the patients on the far left with the highest mutant huntingtin levels are the ones that received placebo, and then as we increased the dose of the drug, the huntingtin levels went down. So the drug was doing what it was supposed to be doing, and we were confident about that because we had this biomarker. So without this biomarker, we wouldn't have known that our drug was doing what it was supposed to be doing. And this might sound like it's a bit trivial, but it's not. This is a really big deal in drug development because even though we have these lovely animal models, they're still always models. And until you go into the patients, you really don't know what you're going to get. And so having these measurements that give you some confidence are really pivotal for the programs. Okay, so one of the other things that we were able to do then is say, look at the level of huntingtin reduction and what does that mean? Do we think this is going to be clinically meaningful? Do we think we're going to have a benefit? And the answer is based on what we had done in the animal models, we knew what kind of level we needed in a tissue to have a benefit in a mouse, and then we can ask, did we see that in our patients? And the answer is yes. So in the CSF of our patients, we had levels that would be above what we would find in a mouse that made that mouse better. And so now we just have to ask, do our patients really get better? So that's really the next phase. And so one final thing that's really important about biomarkers is not only do they give you ideas about across patients, so a patient who received the drug or who didn't, or the different levels of drug, but within a patient, you can watch at how things are happening over time, and you can watch how the drug is affecting them over time. And so here what you're looking at is individual patient levels of huntingtin from each one of those monthly draws, and how it's going over the time as the drug is accumulating, and then the kinetics of how it's going down. So we can really follow in individuals how the drug is lowering their huntingtin protein. Okay. So back to our model. So as I mentioned, we do all this modeling up front to say what our biomarker means, but then at the end we say, was our model right? We predicted our dose and what was going to happen. And when we take that model that our modeler's done and we overlay the data from our clinical trials, it's strikingly accurate. So we were very excited about this. So those dots are individual patients, and that green spread is a spread that would have predicted from the monkeys. And then what we can also do is we can take this now, and since we're pretty confident that the model was accurate since it predicted where they should be, whoops, did I jump it? Oh, then we can go back and predict what that meant for the tissue levels from those patients. And then we're back to those levels are what we think are within the range of what should be clinically meaningful. Okay. Now the next part of this is, so the phase 1 is successful, the drug seems safe, it seems like we're moving it. The next thing is, can we design a pivotal trial? So can we design a trial? Do we know enough about how the disease will progress to say these are the endpoints we want to look at within a clinical trial to see a benefit? And that's, again, where lots of really beautiful work from the Huntington community has come in. And the Huntington's community has done a series of natural history studies. And these studies are really amazing because what they are is they're basically clinical trials that patients are volunteering for, knowing they receive no benefit from the trial itself. They're just giving of themselves and of their data so that we can understand the disease, so that when treatments are ready in the future, that we'll be able to understand it enough to do the therapies. And so this is a lot of work, again, that was led by CHDI and a number of physicians throughout the world following the patients and seeing and really trying to quantify the disease. Another thing that the community has done, and again, this all goes into trying to pick endpoints for that pivotal trial, is looked at additional disease biomarkers. And additional disease biomarkers that can be measured in the fluid. And so as I mentioned, this is work where you can see mutant huntingtin levels go up with disease in the green, and then this is another protein that also goes up with disease. And both the cerebrospinal fluid as well as this protein goes up in the plasma, so in the blood that you can measure as well. So now we have a marker that we could potentially measure in the blood that's going up in the disease. This particular marker is neurofilament. It's a marker that's found in neurons, and as neurons die during the progression of Huntington's disease, they release this protein, and then we can measure it. And so now we have another marker that we can look at, and a marker that we can look at less invasively because you can look at it in blood and see how that's happening and how that's changing to really help us inform how the trial is progressing. And so this is where we're at, and I've highlighted the parts where the biomarkers and the natural history studies really help that timeline. And so this is a really exciting point for us, both in terms of this particular program, because the phase 3, that pivotal study, will be starting later this year, but also in terms of our understanding of what we need to do and what we need to understand, both to develop a drug for the CNS. So these are long timelines, but we've learned a lot, so we can shorten them, and also where to focus our biomarker efforts and strategies, because what's been successful before. And so then just to go back to that initial clinical trial, what we've been able to do here is, because we were able to answer, do we have the dose right, and do we know the endpoints from both that biomarker data as well as the natural history studies, we can skip the phase 2. So we can skip the phase 2 and go right from our phase 1 into a pivotal trial. And so that's how, really a real life example of the importance of investing in understanding your disease, quantifying your disease, and then understanding and trying to find these biomarkers that can help accelerate things. And then finally, just one more summary is these biomarker strategies are really based on regulatory strategies and what you want to do clinically and trying to tighten those timelines. And the real important steps that you need to understand for a biomarker is, first, what assay are you going to use to measure it? How does that move in your patients and move with your drugs? And then finally, do you have a really well-validated assay that's very quantifiable and very precise? And so with that, I think that's everything. And this is just the short list of folks, but the number one is our patients who participated in the phase 1 study, and then the investigators who did that again, and of course our colleagues at CHDI who work closely with on these biomarkers. I think we're ready for questions.
Okay, we have a few minutes for questions. I'll bring you the mic. Please raise your hand.
The data's awesome.
Yeah, that's cool. Thanks.
Hi, I'm Donna Bernstein, a genetic counselor. I'm just wondering, out of curiosity, is there any impact of the number of trinucleotide repeats on the required dose or response to it or efficacy, or is it just anybody who's over the threshold gets the same dose?
Yeah, so that's an excellent question. So just for everybody in the audience, so Huntington's disease is caused by a CAG expansion, and different patients have longer expansions. The longer the expansion, typically the earlier the onset of the disease. So in animal models, our oligonucleotides, they don't target the expansion, they target downstream. So we've done extremely long expansions and extremely short expansions, and then same things in cells as well, a range of expansions. And so we don't see a difference in response preclinically based on CAG repeat expansion length. Now, that said, clinically, we've only done a small subset of patients, and we'll see how that pans out, but none of the data in either cells or animals would predict that.
Hi, great talks. Thank you so much. I lost track. Were there any biomarkers discovered before? When was the CSF discovered? Wasn't that part of the trial?
It was part of the trial.
Yeah.
So, yep, the mutant huntingtin, the CSF was a few years before the trial.
Okay, and they had natural history studies going on for years before that without the biomarker work, but that biomarker was able to be incorporated right in as soon as it was discovered?
Exactly.
So
it was discovered well enough in advance that we were able to do the preclinical work to model it and do the validation for it and include it in the trial.
did that create, in terms of the timeline, did that create a pivot in what had been anticipated for the timeline?
It didn't, because we knew it well enough in advance of when we were still finding the final molecule. But what's happening now is you saw that long timeline for Huntington, so there's a huge learning curve for us, but that's getting shorter and shorter and shorter and shorter. And so as the timelines for drug development are getting shorter because we're understanding our technologies better, and that's not just true for us, that's across a lot of these platforms, the time we have to understand the disease is getting shorter. And then also the time we have to develop these biomarkers are getting shorter. So what we're finding is earlier in the process of when we start thinking this is a target we're gonna go after, that stuff needs to happen. And so then that's the encouragement that I would have, is that try to start those efforts as soon as possible, because the drug development process, at least some of the preclinical stuff, is getting faster as we learn more and we get better at this.
I was also wondering too if there is already an established way to query the patients to see how, if they're having any experiences, different experiences that correlate with that CSF reduction.
Yeah, so the clinical trials are really controlled in what happens, and rightfully so, because you want, you don't want to bias the science or the patients or the doctors in any sort of way. So right now, all that's done under proper tight protocols, but later on, you'll get more of that once things are opened up. Does that make sense?
Yes, I just kind of wonder if sometimes in those, there are any ways that they're measuring, even in early stages, if there's ways that they are measuring
patient-reported, I guess maybe adverse events are being reported, but any of the positives, sounds like no, you can't really do.
No, they do. And so we also have, so in those earlier trials, you include a lot of the things that are going to be endpoints in later trials, as much to understand about the natural variability, but also in how to execute those sort of things. So we have that data as well. And we've shown publicly, I didn't include it here, but that there is a correlation between a positive response and this, but it's so early, it's so preliminary, we really can't conclude anything until the pivotal trial with the proper numbers of patients. But everybody's really enthusiastic about that trial getting started.
It typically is part of traditional drug development. So at phase 1, you're doing dose finding and you're doing safety. And in phase 2, you're continuing safety, but you're trying to see whether or not there is any movement of the endpoints that you care about. And you may be surprised and think that the endpoints you thought were important end up not being important, and so that actually influences what you do in your phase 3 trial. So there's a constant evaluation of the data as it rolls in to try to have the most informed development plan possible.
I'm curious on your thoughts, either of you, on what to do in cases where there may be biomarkers available, which could be used to get the benefits that you talked about, but doing so would involve some pretty unpleasant treatment of a patient. I mean, it may be an MRI or a muscle biopsy or CSF where you're not in the lumbar already. Right. And where is that balance between what you can expect from a patient, often a child in a trial, right, and what would bring down and get the benefit of accelerated development?
Right. I think that's a great question. There's a new guidance that just came out from FDA about 9 or 10 weeks ago, specifically for solely progressive diseases for diseases that are pretty rare. They're in small populations. And one of the things that it says in that guidance is that you need to be able to show, as for looking at substrate reduction in diseases that have enzyme deficiencies and there's an accumulation of some sort of substrate that then ends up harming cells in some way. And one of the things that's in that guidance is it is required, or at least it is asked, that you show at a histological level reduction of the substrate, which as a physician, I hear biopsy. Now, if you're doing anything in the central nervous system, brain biopsies are off the table, or at least they should be off the table. So I think then from the developer standpoint, it is our duty to come up with surrogates of that surrogate that might be predictive. So for instance, CSF, cerebrospinal fluid, might be a good surrogate of what's going on in the tissue. And as Holly was showing, you can leverage preclinical data where you can use animal models to show that there's a good correlation between what you see in a more accessible fluid or a more accessible tissue like a fluid than you do in the actual tissue itself. And if you can move them as they have done into something that you could get out of blood and show that it's correlated, that's the way I think that we have to try to approach that. In general, we try to be as non-invasive as possible because the last thing you ever want to do is expose patients to potential new harm that they wouldn't have been exposed to in the first place. It's a really good point.
Okay, well, time's up. Thank you, Steve and Holly, for sharing your expertise with us today. Let's give them a hand. Just housekeeping notes really quickly. At this time, before heading out to the networking break, we would appreciate if you could jump into the mobile app and take our brief survey on this session to provide feedback. Our next breakout session will begin at 4:15, so in 15 minutes. We would like to thank our networking break sponsors, Audentis. Check out our map and agenda feature on the mobile app to see where the next sessions are, or ask a Global Genes volunteer or staffer. Thank you.